Okay, welcome back. We are going to finish up talking about neurotransmitters and talk about neurotransmitter receptors. And that will sort of wrap up our review, sort of advanced information about neuroscience. And then next week we will dive into Sapolsky's book and start our content there. So, I want to start today by going back one slide from the last lecture where I was wrapping up talking about glutamate, because glutamate and GABA are interlocked, so to speak, with each other in terms of synthesis pathways. So, just to refresh your memory, glutamate synthesis happens in the axon terminal, and the precursor for that is glutamine. And so here is glutamine, GLN, which is synthesized in the glial cell and then transported to the axon terminal, where another enzyme modifies glutamine into glutamate, and then glutamate can be released. All right. So in talking about GABA, which is the traditional sort of inhibitory neurotransmitter, there are two primary receptors, and the synthesis for GABA comes from glutamate, and we'll talk about that on the next slide. But two primary receptors that are ionotropic. So we have a GABA A, which is going to open a chloride channel and allow negative chloride ions to enter the cell, and that's how it produces its IPSP. Or GABA B, which mediates potassium channels, allows positive potassium to leave, to exit the cell, which is going to produce a negative charge and result in an IPSP. All right, so here's the pathway. So we talked about glutamine being released by astrocytes and then taken up by now a GABAergic cell, which is then going to convert glutamine into glutamate, and then glutamate gets converted into GABA, and then GABA is released. So the primary deactivation is reuptake through the GAT transporter protein here in the axon terminal, or absorption by glial cells, and then that GABA can be recycled in both cases. There also is some enzymatic breakdown, but the primary deactivation is reuptake. Another more specific inhibitory neurotransmitter is glycine. And glycine is used to help control motor activity in the brainstem and in the spinal cord, sort of to regulate descending motor signals. It has an interaction with GABA receptors and may be co-expressed on the same neurons. So you could have neurons that are being inhibited by both glycine and by GABA in different capacities. All right. At the level of our spinal cord, on the descending motor pathway, there's an area rich with glycine receptors. And when you are asleep, this pathway is activated so that you're essentially paralyzed. The glycine sort of paralyzes yourself so that you don't have any muscle movements during sleep. And that is why when you have REM sleep, the cranial nerves that control facial and eye movements are above the level of the spinal cord where this glycine inhibition is happening. So you do have movement of the face during that REM sleep period, which is why it's called rapid eye movement, why you have that. Strychnine, the poison strychnine, is an antagonist for glycine. So it's going to block the ability to regulate these motor signals, which allows the motor system to go unchecked and results in overstimulating or muscle spasms that can be paralytic. And that's one of the ways that strychnine can kill you. All right, the biogenic amine family. So this involves the catecholamines and indoleamines. The catecholamines is the family of dopamine, norepinephrine, and epinephrine. Norepinephrine is also sometimes referred to as noradrenaline, and epinephrine is sometimes referred to as adrenaline. So if you see those in Sapolsky's book, don't be confused by that. These three neurotransmitters are grouped together because they all come from the same synthesis pathway, starting with tyrosine. Serotonin is the indolamine, and it has a tryptophan precursor protein. Serotonin involved in circadian rhythm and emotional states. All of these biogenic amines, the catecholamines and serotonin, typically are, well, dopamine and serotonin are definitely neuromodulatory in nature. They can be used as discrete signaling molecules like dopamine in the reward system, but in general, they're sort of affecting the excitability of other neurons. Histamine is also in this group. Histamine comes from histidine precursor protein. It is involved in our sleep-wake cycle in the wake portion of that, which is why an antihistamine can cause you to become drowsy. But it's also used in the peripheral nervous system. Histamine is released in response to tissue damage or irritation, such as allergies, which is why antihistamines are good for when you have a cold or you have allergies, but not so good in the brain. So good in the periphery, not good in the central nervous system. The once-a-day allergy pills like Claritin or Allegra are antihistamines that have been engineered to not cross the blood-brain barrier. So they can produce their effects on the irritation in the peripheral nervous system, but won't produce the drowsy side effects in the brain. All right, so looking at these synthesis and removal pathways for dopamine and norepinephrine. We have this common pathway from tyrosine here, which gets converted into L-DOPA, and then from L-DOPA converted into dopamine. And if this is a dopaminergic neuron, that's where the process stops. Dopamine is packaged into vesicles and can be released. However, if it's a norepinephrine neuron, there's another enzyme present that turns dopamine into norepinephrine, and then the norepinephrine can be packaged and released. Right? Okay, so that enzyme is dopamine beta hydroxylase that converts it further into norepinephrine. Major source of norepinephrine in the brain is the locus coeruleus, long diffuse neurons that go throughout the cortex to induce arousal in the brain. Removal primarily happens through transport, reuptake, and metabolism, a metabolizing enzyme. So VMAT is a particular transporter, and NET is another transporter that you will see, and that metabolizing enzyme is called COMT, catecholamine methyltransferase. And so those are the two primary ways that dopamine is removed from the synapse. These are also targets then for drug therapy if we want to increase dopamine, where we can block reuptake or block the enzymatic breakdown. Serotonin, as I mentioned, is synthesized from tryptophan. It actually goes through a two-stage process to get to 5-HT, which is serotonin. Serotonin has a major source of release is the Raphe nucleus, also found in the brainstem, along with locus coeruleus. And again, serotonin is neuromodulatory. There are diffuse projections throughout the cortex. Primary breakdown for serotonin is similar to dopamine and norepinephrine in terms of reuptake through the SERT transporter, serotonin transporter protein, for recycling, and enzymatic breakdown by the monoamine oxidase enzyme. Again, both of those are targets in terms of increasing serotonin, as we'll talk about when we talk about major depressive disorder, with SSRIs, selective serotonin reuptake inhibitors, so drugs that are going to block the ability to reuptake serotonin, or MAOIs, monoamine oxidase inhibitors, that are going to block the ability of MAO to break down serotonin. All right. One particular example of how serotonin can have a neuromodulatory effect is shown here, where serotonin can have an axonal-axonic, axon-to-axon connection through a second messenger, through a metabotropic receptor, activate a G protein, which causes an increase in protein kinase A, which can then close a potassium channel. And when we close a potassium channel, that is going to increase the positivity in that axon terminal. And so now when an axon action potential arrives at the axon terminal, it's going to be higher than its normal baseline resting potential, which is going to allow it to become more positive for longer, allow greater calcium influx, and then allow greater release of neurotransmitter. All right. I want to talk briefly about nitric oxide because it's important in learning and memory. Nitric oxide is a gas, not to be confused with nitrous oxide, which is an anesthetic gas, but this is nitric oxide acts as a signaling molecule. It influences one of those second messengers, cyclic GMP, and that then decreases the production of glutamate. And so in the LTP, AMPA, NMDA learning circuit, nitric oxide after LTP occurs, is generated and released, diffused back to, as a retrograde signal, back to decrease the further release of glutamate after learning. All right, so there are lots and lots and lots of neurotransmitter receptors. We talked about kainate, quescolate, non-MDMA, metabotropic glutamate receptors. That's these down here, class 1 and 5, class 2 and 3. But you can see that there are lots and lots of variants or subfamilies of these receptors. So what does that mean? It means that all of these 5 receptor families here can be activated by norepinephrine and epinephrine. But then what happens when they get activated is slightly different, or where these receptors are located is in a different place. So for instance, the beta family of norepinephrine receptors are found in our breathing passageways, and are activated in response to constrict our airways. So people that have asthma and their airways become constricted can take a beta blocker in an inhaler, inhale that beta blocker, and it will block and cause relaxation of those airways. It would not act, but that drug is engineered only to act on the beta receptors and would not act on alpha norepinephrine receptors. All right, so there are 4 general classes of neurotransmitter receptors. There's ionotropic that we've talked a lot about, where the neurotransmitter, it's a neurotransmitter gated channel. Neurotransmitter opens the gate and ions are allowed to flow in or out of the receptor. But we can also have enzymatic receptors, in which case an enzyme is produced. It's sort of similar to a metabotropic, but it does not have a G protein coupled component. It has an enzyme component on the inside. Metabotropic receptors, also referred to as 7TM, 7 transmembrane receptors, because they actually go in and out of the membrane 7 times. This is not a very good cartoon representation of that, but they have a binding site on the outside, and then they have an activation terminal on the inside that has a G protein coupled to it. So sometimes you'll see these also referred to as G protein coupled receptors, or GPCR. When the neurotransmitter binds, it activates that G protein. We'll talk more about that in a minute. And then there can also be intracellular signaling. So there are some signaling molecules like the steroid hormones, estrogen and testosterone, that are able to travel through the lipid membrane excuse me, and actually activate receptors intracellular receptors inside the neurons. Okay, so as you know, these are sort of simplified representations of these types of receptors, and what you really need to understand is that it's very, very complex. So take estrogen, for example. I mentioned it can enter through the lipid membrane and it has intracellular binding sites. However, it also has G protein coupled binding. It can also act on metabotropic receptors, and so here's a metabotropic receptor, and it can have enzymatic binding. So here we see that 7-TM, where there's 7 membranes through. You have your N side, your N terminal, which is going to bind to the estrogen, and then you have your C terminal, which is going to have your G proteins coupled to it. And then it can also act as an enzyme-linked receptor. So here we see that. There are lots of, not only can the signaling be complex, but what happens after the signaling is very complex as well, and results in various different types of behaviors that you see represented here. All right, so let's talk a little bit more about G proteins. G proteins are, we call them neuromodulatory because in general they're not doing discrete cell-to-cell signaling, but they're changing the excitability, right? So in this particular example, GDP is the G protein coupled receptor, which gets phosphorylated into GTP. The GTP can then go open an ionotropic channel from the inside, which is going to allow, in this case, potassium to exit. So this is going to decrease the excitability of that neuron. You can also have other activations where you can have GTP activate an enzyme, which is going to then produce cyclic AMP. Cyclic AMP can then go act on another protein kinase, like protein kinase A, and which then could do lots of things. It could generate more channels. It could open an ion channel. And so this process could have the same outcome as acting directly on the ion channel, but typically has a longer lasting effect because we've amplified the signal at both of these steps. And then finally, another benefit of G protein coupled receptors or metabotropic receptors is that this enzymatic pathway can go act on the DNA to actually create new proteins, new channels, more neurotransmitter receptors. Again, all sorts of things that change the excitability of that cell. All right, so why have these multiple sequences? Well, at each step of this system, you get amplification of the signal. So a G protein that's activated to adenylate cyclase, that G protein may activate 10 adenylate cyclases. Each of one of those 10 adenylate cyclases may activate 10 cyclic AMP molecules. So now we're up to 100 cyclic AMP molecules, which in turn then could act on more protein kinase A. So if each of those 100 cyclic AMP act on 10 create 10 protein kinase A, now we're up to 1,000, right? So we get a small one neurotransmitter G protein activation resulting in a very big signal sequence due to this cascade. So some important ones you'll see: cyclic AMP. Cyclic AMP can have the same pathway that results in 2 different processes depending on how that neuron recognizes the end product protein kinase A. You can also have divergent pathways. So here phospholipase C can activate both IP3 and diacylglycerol, which can activate protein kinase C, and calcium release from IP3. Both of those then can result in changes in the cell membrane structure. And so in LTP, when we're adding additional synapses, when we're adding other receptors, we are activating pathways similar to this. All right, so as I mentioned, this is way more complicated if we get down to the specifics of it than just like 2 or 3 signaling molecules, right? So we don't need to know all of this at this level, but just be aware that there are lots and lots of different signaling pathways. All right. Last thing, we'll talk a little bit about exocytosis, a little review for you that vesicles are full of neurotransmitters in that active zone of the synapse, and they are bound to the to the inside of the cell membrane through a complex called SNARE. It consists of 3 different proteins. Synaptobrevin is on the vesicle, and it intertwines with syntaxin and SNAP25 to dock the vesicle. The third protein, synaptotagmin, is sitting there waiting for an influx of calcium to cause the 2 membranes of the vesicle and of the cell membrane to fuse and release the neurotransmitter. So when we have an influx of calcium that now comes in through a voltage-gated calcium channel, we activate synaptotagmin, it fuses the membrane and allows the dumping of that. Once this process is over, another molecule called dynamin is going to sort of pinch off the cell membrane from the vesicle from the cell membrane wall and recycle it, allow it to go be refilled and redocked. So again, things are more complicated than just this, but what we see is that we have in an axon terminal an active zone here, and then we have a whole bunch of full and empty vesicles. So the active zone, these are all docked and waiting with the SNARE complex, and the ones in black are filled vesicles waiting to take their spot here, and then the gray ones are in the recycling pool, so those are waiting to be refilled. All right, so a little bit about ways in which we can manipulate this process of neurotransmitter release. So Botox comes from the botulin toxin. It's a refined form of this toxin. And here you see a before and after. And so Botox essentially paralyzes muscles by preventing the release of acetylcholine at the neuromuscular junction. So after injecting in these areas around the eyes, the muscles relax and the skin becomes more smooth. Here we see where the muscles are tense. Here we see afterwards where they've been paralyzed and by Botox, and they're smoother. Lasts about 6 months, and then you have to have it redone. And its mechanism of action is it prevents acetylcholine from being able to bind at that at the neuromuscular junction. So it interferes with SNAP25 so that the vesicles can't effectively bind to the terminal. Right? Another toxin, tetanus toxin, also activates, also interferes with this docking mechanism, but it interferes with synaptobrevin to prevent, again, neurotransmitter from being able to dock. So tetanus toxin, when you step on a rusty nail or have a cut and tetanus enters the body, it actually travels retrograde through the alpha motor neuron into the spinal cord, and it affects this inhibitory interneuron right here. This neuron is important in our proprioception. So we have Golgi tendon organs here that detect tension. So when do we have tension in our muscles? When they're contracted. So this 1B sensory neuron is sending a signal, hey, there's tension in the muscle. I'm going to activate this inhibitory neuron, which is going to inhibit muscle contraction and allow the muscle to relax. Well, if we knock out that inhibitory interneuron with by having tetanus toxin interfere with synaptobrevin here, so that inhibitory neurotransmitter cannot be released, then we eliminate this feedback signal. And so now the muscles are very tense. All the muscles are tense here, as shown in this picture, and we have no mechanism to relax or stop sending those excitatory signals. Okay, So that is your mini Friday lecture. Get out of class a little bit early. I am very, very much looking forward to seeing you in person Monday at 10:30 in our classroom, which I believe is 225. Not exactly sure if that's correct, but check the syllabus and make sure. And have a good weekend. And here's your Ed puzzle question.